Yeah, listen, good evening, everybody. My name is Heng Lin. I'm based in Melbourne. I'm on this tech team, and I'm originally from Malaysia. Um, I will be talking quite broadly here on some of the challenges, future opportunities in population genomics, with a small example on one of the things that we have worked on previously in rabbit gene drive in Australia. First, I'd like to begin by acknowledging the Wurundjeri Woi-wurrung people as the traditional owners of the lands that I am presenting from today, and pay my respects to their elders past and present. Just a quick outline. I'll start off quite basically with population genetics, then talk a little bit about challenges, and then on future opportunities and an example. Okay, so when we analyze population genetic data, we are actually studying one or more of four major forces that shape population genetics. So the one that the major ones mutations. So we can break this into serotypes. That's SNP or SNPs, single nucleotide polymorphisms, which are variation on a single DNA base. Then we have structural variation ranging from single base insertion into the genome or deletions, or up to very large rearrangements and movement of chromosomal pieces. There are many ways to detect mutations. These days, we just do sequencing. Previously, as Tech has presented earlier, there are things like microsatellites as RFLPs, etc. So next three forces are all measured over time, and they shape population genetics by affecting the frequency of the variation in a given population. So we have gene flow, the movement between subpopulations that allow variations to move around directly, and that thus affecting the frequency of the variation. There are gene flow analyses that range from kinships to migration analysis. And in the context of pest populations, gene flow analysis can highlight hybridization between species, movement of resistance, or possibly diagnose origin of pest incursions. Then we have genetic drift. That's practically just stochastic or random error. That's dependent on population size. So frequency of variation alleles more likely to modulate over time if we have smaller populations compared to that of larger populations. And then coupled with gene flow, it can give us an idea whether invasive pests established from a small founding numbers or larger ones or even multiple introductions. And finally, the fourth force is selection, which is the modulation of frequency of the variation or alleles due to change in in the environment that preferences one variation over the other. So things like pesticides, climate change can affect, can select for certain variations, and these are of interest to look at. Over the last 15 years, the sequencing technologies to uncover the genetic code of individuals and populations have been ever improving. We have gone from PCRs of small number of genetic regions to whole genomes, from short reads to long read sequencing. And now we have far greater volumes of data, so allowing us to answer more complex questions. So here's just a non-exhaustive list of some of the challenges. I'll start with the most important one, and Tech has highlighted it quite well already. So data complexity and data quality. So the amount of markers you have and how you design your sampling is really crucial. because error and bias in the way you sample can significantly impair the conclusions that we make. We have reference genomes. So having a reference genome is extremely crucial for studying populations of pests. One problem with not having a reference genome and using another species reference genome is that we potentially lose crucial information in regions of genome that do not match between species. And we don't get a full picture of what is happening to our study species across their populations. I mean, as an analogy, we can say a reference genome is like a reference book. And then let's say we have taken similar books and we've torn them up and we want to piece them together. We could use this reference book to piece them together. But if we use a completely different book, so a different species, then some chapters might be missing and we might be putting together pieces that may not ever be found in the reference book. And it can't be resolved and then it ends up being lost. And so we're going to have loss of some information. We have, as we've shown earlier, the volume of data has increased significantly over the last decade. We need significant amount of storage space and computing power just to deal with the amount of data. We also need to improve efficiency of our methods. There's also a mind-boggling number of methodology out there, which can make it quite difficult for newcomers to navigate. There's a need to have more integrated processes that can reduce all the time wasted trying to just make things work. So here's an illustration from just a human genome research alone. And they anticipate in the next decade there's going to be 40 exabyte of data. That's 40 and 18 zeros behind it. So if we think about everything else, including pest management and conservation genetics, there's going to be a lot of data out there. And with that, also we have cost of genomes are decreasing significantly in the context of human genomes. The 3 gigabase of genome it started off from 100 overs of millions at the beginning of the Human Genome Consortium to now it's like below $1,000. There are other challenges I will quickly highlight. One of them is resolving complex variants. So looking at large structural variations in genome, so indels, deletions, those are still very difficult to analyze, except in the context of if we have long read data. In a pest management perspective, we also have problems with hybridization causing closely related species like in Helicobacter, which could have implications on which it could, as Jen Peng said, creating a mega pest. The other challenge includes also like monitoring evolution and the movement of resistance. And finally, I also wanted to highlight like pest management is quite a global problem and not a country by country problem. And sometimes communicating population genomic results can potentially cause geopolitical tensions because, and so it's just no benefit if we play the blame game. Rather, it'd be good if we can work together to tackle the problem. And last but not least, one of the challenges is manpower, so people and expertise. It takes time, money to train people up. So we need to not just have more resources to train people, but we also need to have more sustainable approaches that we will allow people to get up to speed so that they can concentrate on the most important parts of the research, which is the analysis and interpretation of results, rather than the less mundane things like management of data or figuring out how a program works. So with that in mind, the Applied Genomics Initiative at CSIRO was formed, led by Dr. Rahul Rane and Dr. Tom Walsh. with the core idea that scientists should, in the future, be able to not spend so much time on the complexities of data management, navigating complex workflows, but rather analyzing, interpreting, and making decisions, helping us to get to the end goal faster. So here I made a little I use AI to generate this figure, and I realized that for future perspective, I realized after listening to the talks, I forgot to include a multi-omics integration because the parts like transcriptomics, proteomics, they're all really important part of the puzzle of piecing together the biology of tests. I also noted here CRISPR-Cas gene drive as an upcoming potential, although this should not preclude all the existing biocontrol methods, which already heavily depend on population genomics to guide decisions on how to deploy biological controls. We then also have trying to integrate ecology and spatial data for monitoring and controlling pests, and also the development of more precise resistant management, and all of it in leveraging, hopefully, the advancing large language models, which can help aid research in making quicker decisions. With the ever-growing data volume and methodology, there's a need for people to band together to create frameworks such as standardized repositories and protocols so that we can leverage the new age of technologies and enable efficient science and better accountability. So for example, in farm mealworm or brown plant hopper and other pests, from time to time, it'd be great to come together, put together, collate all the genetic data from across the Asia Pacific and beyond, incorporating previous studies, and do a comprehensive review using the most up-to-date sequencing technology, and so that we can see whether we can resolve some of the debates that have been ongoing and hopefully help us to move towards solving the problem at hand. I'm going to skim through quickly on an example in gene drive. So over the last decade, we have CRISPR, which have revolutionized gene editing through introduction of Cas9 enzymes and specific RNA sequences, like the one in yellow is, that is complementary to a gene region, so that you can achieve double-stranded breaks, which then leads to mutation of the gene of interest. So the mechanism of this Cas9 RNA sequence binding is quite sequence specific. So a mutation in anywhere in the gene can actually cause a resistance to Cas9. So a few years after Cas9 became global news as an engineering tool, somebody in the US thought of, why not if we introduce Cas9, CRISPR-Cas9, to target a gene, but also can be inherited at the same time? Then it can be self-propagating and also then edit the genes in wild population. This is what we call gene drive. Gene drive is not new, and there are lots of natural ones out there. But using CRISPR-Cas9 is actually a new idea, at least in the last 10 years. So usually we get 50-50 inheritance from parents to offspring, but in gene drive, we can get up to 100% of inheritance. So say if we introduce a mutation that causes all the offspring to be male, then eventually all the offspring will become male and then the population will crash. So you can see that this could have potential risks to populations that we don't intend to affect if gene drive spreads around. Given that populations of organisms, including have some level of genetic variation, there's a need to understand how genetic variation can impact gene drive. Together with Dr. Rahul Rane and Owen Edwards, we created GUARD, the Gene Drive Utility and Risk Determination Platform, and we set out to streamline the process, which includes analyzing population genetic data in identifying candidate regions for gene drive through use of some sequence-specific rules and population dynamic models that takes actual sequence data to infer efficacies of gene drive models. These models came from collaborators from Melbourne, University of Melbourne. And a few years ago, we had an opportunity to test this in rabbits, this framework in rabbits, a major pest in Australian landscapes causing billions of dollars of damage, but it's endangered in native range in Europe. So the idea with rabbit biocontrol is we want to suppress rabbit populations in Australia, but not really in Europe. So we set out to identify whether we can find some candidates specialized to Australian rabbits. So we sequenced quite a lot of rabbits, Australian range, and together with collaborators in Europe, we also obtained sequences from them in a separate study for European rabbits and also other Australian rabbits. Coming to the main challenge again, so where data quality. And so because we have multiple sequencing initiatives, we encountered challenges with data compatibility, notably sequencing depth differences. Here's a PCA. So PCA is meant to separate out multivariate data into axes of most variation. In population genomics, the hope is that major population differences will separate out in PCA, with distances reflecting genetic divergence. On the left was our initial analysis of the combined datasets from our study and the European colleagues. The dark green was our European colleagues' data, and the rest was ours. And so you can see it's a huge batch effect there. So luckily, we have common samples between the two datasets, which allowed us to notice this problem. And fortunately, we will manage to mitigate this through careful, justified filtering to uncover the pattern that observed in on the right. Coming back to identifying targets, we actually managed to identify lots of targets. So over 100,000 targets, 2,000 of them are within 1,300 genes, and about there's potentially two that could be of interest. These candidates are guaranteed to work if we are in a perfect scenario for gene drive. So like the one I showed here in the simulations, where blue is the gene drive frequency and the red is the wild type variation. So just to summarize, our experience with this data further iterate the data complexity challenge. Ideally, we should mitigate early on, but there should be some fail-safe measures to allow data to be salvaged so that we can avoid making the wrong interpretations. On the gene drive perspective, we have shown that we have some bioinformatically identified candidates, but it's still early days because we still need to have laboratory proof of concept that this thing can be engineered into rabbits. I didn't talk about regulatory, but we were also together with Rahul, and we were developing the dashboard which collates all this information. So hopefully that we can give regulatory and ethical bodies a chance to play around with the data and decide what risks are acceptable. And I would like to finish off just to reiterate that the main challenge in data complexity. Before we actually embark on expensive pursuits, we need to make sure there's clarity in the question we ask, and thus there's purpose to why we are sampling. And hopefully that also means we sample strategically and not aimlessly. Because if we don't, we might end up having a mess later on. Thank you.